Hello friends, my name is P. Balaji, Department of MSc Microbiology in Sacred Heart College, Tirupattur. Now we are going to see about algal biofertilizer isolation and mass cultivation of blue-green algae. Introduction. Biofertilizer are natural fertilizers containing living microorganisms that enhance nutrition availability to plant. Among them, algal biofertilizers, especially blue-green algae, also known as cyanobacteria, play a vital role in agriculture. This microorganism can fix atmospheric nitrogen and convert into form usable by plant. A well-known symbiotic association is Abonia azolei living in the water from azolla which enhance soil nitrogen living in rice cultivation. Importance of blue-green algae in agriculture. Blue-green algae contribute significantly sustainable farming by enriching soil fertility through biological nitrogen fixation. They improve soil structure, organic matters content, and water holding capacity. Being eco-friendly, they reduce the dependence on chemical fertilizers, lowering both cost and environmental pollution. Additionally, they promote better plant growth, higher yield, and help maintain ecological balance in agriculture ecosystem. Common genera used as a biofertilizer. The most commonly used BJ as a biofertilizer protections are Anagonia, Nostoc, Alsoria, Toilportix, Cylindrospermum, and Calotryx. These genera are known from their efficient nitrogen fixing ability through specialized cells called heterocytes. They thrive in moist soil and standing water, making them ideal for rice field and wetland agriculture. Isolation of blue-green algae. Sample collection. Samples are collected from natural habitat like rice field, ponds or wet soil using sterilizer containers to avoid contamination. Media preparation. Blue-green algae can be cultured in nutrient media such as BG11 medium or Alline and Arnon medium. BG11 medium is commonly used and contains essential minerals, while Alline and Arnon media is prepared for nitrogen fixing species as it does not include a combined nitrogen source. Isolation techniques. The collected samples are processed using methods such as serial dilution, agar plating, on stick planting to obtain pure culture. These plants are incubated under controlled light and temperature conditions. Pure colonies are identified and selected using microscope examination based on their characteristics morphology such as filamentous formation, presence of heterocytes and actinides. Incubation condition. Culture and maintained at a temperature of 25 to 30 degrees Celsius with 12 to 16 hours of light exposure daily. The pH of the medium should be between 7 and 8. Visible growth typically occurs after 10 to 20 days. Mass cultivation of blue-green algae. Field method. For large scale production, open sunny field and selected. The soil is leveled and poles of about 10 meter by 2 meter are prepared with raised bunds to hold water. Each plot is folded with 3 to 5 centimeter of water. To promote growth, superphosphate and slurry of soil or cow dung are added in nutrient courses. The plot are then inoculated within 5 to 10 kg of PGA culture per acre. Water is maintained at a shallow level throughout the growth period and the culture is allowed to grow for about 10 to 15 days. Once a thick algal mint development on the surface, it is collected, dried in shade and stored in polythene bags for future field application. Trout method for small scale or laboratory level multiplication, cement or plastic through are used. This through are fixed within a mixture of soil and water, and a small amount of superphosphate is added. After incubation within a blue-green algae culture, the two are kept under natural sunlight for about 2 weeks. The grow algal mat is harvested and dried similarly. Application in field. The dried blue-green algae biofertilizer is applied at the rate of about 10 to 12 kg per hectare in a standing water condition, preferable 7 to 10 days after transplanting paddy. The material is evenly spread over the water surface to ensure uniform colonization. The algae grow and multiply in the field, enriching the soil with nitrogen and other nutrition. Advantages of blue-green algae. Blue-green algae is one of the most efficient, natural and inexpensive sources of nitrogen. They can fix up to 20 to 200 kg of nitrogen per hectare per crop section, reducing the need for synthetic fertilizers by nearly one-third. Apart from the improving soil fertility, they enhance crop yield by 10 to 15%. They also help maintain the physical structure and microbial health of the soil. Begin renewable and biodegradable blue-green algae biofertilizer are safe for the environment and help in reducing pollution caused by excessive uses of chemical fertilizer. Limitation. Although beneficial by blue-green algae biofertilizer have some limitation, they grow best under waterlogging conditions and are therefore mainly useful for paddy cultivation. Their cultivate many reduced by the uses of pesticides of herbicides. Growth is also affected by inadequate sunlight or low temperature. Moreover, compared to the chemical fertilizer, their initially growth is slow and the result take time to appear. Recent development in algae biofertilizer research including the uses of mixed culture blue-green algae with azolla, which enhance nitrogen fixation efficiently. Scientists are working on a genetic improvement of strains to increasing nitrogen fixing capacity and resistance to environmental stress. Encapsulation techniques have been introduced to extend the shelf life of blue-green algae cultures. Integration of BGA biofertilizer with organic and integrated nutrient management practices in gaining polarity and sustainable agriculture. Thank you.